Hi everybody. So welcome to this class for those who watch it in recording. So as you can see in my back, this is my office. We're not having the class in the room because there was an exam that was scheduled at the same time. So today is yet another class online. So before we start about today's topic, there was really a big cut. Like the first 6 classes were about low-level understanding of genomics, which means sequencing technologies, assembly, read mapping, etc. The second half of the course is much more based on the high-level understanding. So there's going to be a real cut in terms of the topics that we're going to approach, but also in terms of where are we going to put our focus in terms of what are the grades that we care for, etc. So the first half was about the midterm, and now this half is about the final exam. So from today, I recommend that you get started if you haven't done so on your final exam. And I'd like to give you a couple of hints about what's going to happen now. So the BIOD25 project is really to create the genomics course that I cannot teach. This is mission impossible, but we still have a shot at it. So what would be nice for a course would be to be able to cover all the topics of genomics, to learn to write like a blogger, to create some online content about genomics, to start building your portfolio, and put UTSC on the map of genomics. So these are all the things that I'm trying to aim for in BIOD25. And I thought that for this, the best is that you practice the real thing, which is to make content that will actually go online. And writing like a blogger is one of the best exercises that you can think of, because it forces you to process highly complex information, high-level understanding, and to simplify it in something that can be understood by people who don't have your knowledge. And that is one of the hardest exercises that you can do intellectually. So basically the point is, let's all write a blog post about something that's fairly recent and interesting in genomics, and that will allow us to produce a course that will go much more beyond what I could teach myself. Because now you are becoming the teachers. You will teach each other, but you will also teach other people in the community. So in a sense, it's a meta course. Like I'm teaching you and you teach other people. So in terms of logistics, it's organized this way. So there are 5 steps. The articles will contain a title, a TLDR, too long didn't read. So that's an abstract, a short version, a picture, actually 2 pictures at least, and even more if you like, and then the content proper of the article. So you already have chosen an article, so that was the easy part. The next milestone on our list is to get a title and a TLDR. And this is going to come quite soon, you'll see. So you need to read the articles now, because at that point already, when milestone number 2 comes, you have to be able to know what you want to write about. Okay, at least give a title and a short summary of your article. So you have to know what you're going to talk about right now. If you haven't done so, read your article in full. Next week, I'm going to give you some hints about how to read an article efficiently, just to make sure that you're not wasting too much time with it. If it takes you more than a week to read your article, it's okay, it's normal, especially if it's the first. It took me more than 2 weeks to read my first scientific article when I was a student in 3rd year. So I don't expect you to be able to just read a paper like that and be able to say what's inside. So it takes long the first time. The good news is that when you have the techniques, when you use the techniques that I've developed over the years, some of them I can share, it takes me now approximately 20 minutes to read an article. So if you can go from 2 weeks to 20 minutes, that's a huge improvement in terms of just techniques to process very fast information in articles. I'm going to say more next week about this. So the 3rd milestone is the pictures. You will have to make or to choose pictures to illustrate your topic in the blog post. And then most of the hard part is going to be milestone number 4, where you actually have to write the content of the article. That's the hard part. And then after you submit your articles, your blog posts, there's the finishing touch in which you have to give feedback to other posts. It's going to be a random matching. You will have several blog posts to read, and you will have to propose feedback, like to give some hints about how the text could be improved. So that's what's going to happen in the second half of the semester. I hope that this is relatively clear for most people. Any questions about the final exam before we move on with today's topic. No questions, so let's go on. So today's class is about variations and, there is a question. To clarify, there is no final exam. Well, this is the final exam. There is no limited time sitting exam. Okay, so that will not exist in BIOD25. It's kind of a final essay, if you want. Okay, so there is not a single exam of BIOD25 in which you have to be in a room in which there's a limited time or you have to do whatever. So why is that? It's because I don't believe that this is a good way of making exams. I don't believe that this represents anything real, like there's any job in the world where your task will be like that. You will never be in a room without access to the internet, without being able to talk to anyone, without having access to your phone, and just having to remember everything and then write it down because people will ask you some questions. The only thing that looks like this, if I have to be fair, is a job interview. So, like, fair enough for people who would have to go through job interviews, maybe it would be a good preparation for them. But for any task that I know in the world, there's none of them that looks like an exam. So I prefer to get you ready for the real stuff. Most of the jobs nowadays, if not all of them, you have more than 2 hours. You usually have weeks in terms of doing whatever you have to do. You have access to the internet, you have access to your friends, you have access to whatever means to do your task. You can work overnight if you want, just to speed up the work if you're working close to the deadline, etc. But most of the jobs nowadays, they tell you that's what you gotta do and that's the deadline. Just do it, goodbye. So that's the way that I think the exam should be. So it is the final exam, short answer. All right, so today we're going to talk about variations and 4 and 6. And so let's get started with what variations are. Like when we speak about genetic variations, what do we mean? So the variations are in the human genomes. We don't have all the same genomes. I think that's kind of obvious. So there is a reference genome, the one that you've been already working with a little bit using BLAST and other tools. But there's just one genome, which is nobody's genome. Actually, nobody has that genome on the planet. We each have a slightly different version of that genome. So how different are we? Several large projects collect the genetic variations in the human population. The databases currently hold 80 to 90 million SNPs, so single nucleotide polymorphisms, and approximately 6 million structural variants. Most SNPs have no effect on the phenotype. For structural variants, the jury is still out. If you remember the first class, so we've seen that there are all these SNPs that appear on the browser, but only some of them are highlighted in the OMIM database. And so OMIM database really traces the SNPs that have an effect on human diseases or phenotype or something like that. And so it means that only a small minority of SNPs are actually causing any visible variation in the body or in the mind. So what are the variations? What types of variations exist? A typical person differs from the reference human genome at approximately 5 million sites. Okay, so that's one every 500 to 1,000 base pairs on average. So if we compare the two genomes, like your genome and the genome in the reference, you'd see that you have a stretch of 500 to 1,000 nucleotides that are exactly identical to the reference, but then every so often you'd have a different letter in your genome. So 99.99% of the variations are SNPs and short indels. Indels, that's insertions and deletions, and short means we're talking 1, 2, or 3 nucleotides. We're not talking more than that. Okay, so 99.99% are really one letter variation, or let's say 1 to 3 letter variation. That's really short range variations. But the rest, like the 0.001% that remains, are approximately 2,500 structural variants. They are sometimes abbreviated SVs, with 1,000 large deletions. And large, we're talking about 1 kb or more, something that's not 1 or 2 nucleotides. 150 copy number variations. So it means that these are relatively large pieces of the genome in which we don't have the same number of copies. I may have 3 repeats in a row, you may have 7. 1,000 ALU insertions. Remember ALU? It's this repeated element that's created by the line, and so we have millions of them. But if you compare my genome and your genome, there will be 1,000 insertions that are different between yours and mine. 125 line 1 insertions. Remember line 1? It's like bigger, like a bigger mobile element, which is 8 KB, and a few other things, and that will represent these 0.001% of the sites that are big size variations or structural variations. This adds up to approximately 20 million nucleotides that differ from the reference. So even though there are much fewer sites that are different in terms of these structural variants, the total number of nucleotides that are different is actually very big. It's something to understand. So this is why the jury is still out in terms of are they important for phenotype and for diseases, because on the one hand there are few of them, but on the other hand the effects are much bigger because they're much larger. What am I doing teaching without my scarf? Doesn't work. So here are examples of types of variations. So one of them is single nucleotide variant. So you can see here this is represented by this little green box here. So you got 3 nucleotides and one of them is different. That's here the SNP. You can have a deletion, so the nucleotides that are present in the reference are gone. So that's this, this arrow that's like that. An insertion, which means that the genome of the person here in blue actually has more nucleotides than in the reference genome. It can be duplications, like something that's present in 1 copy in the reference genome is present in 2 copies in the genome of the person. You can have interspersed duplications, in which, like for example, in the reference genome there's 1 copy, but in the genome of the person there'd be 2 copies, but they're not in tandem. They're interspersed, like it's at another location, possibly on another chromosome. Inversions as well, so the sequence has been inverted like that. Translocation, the sequence has moved around, so this is typically the case of transposons. Remember that they make cut paste, so that would create a translocation of the content. And you have copy number variants, so like here this is a triple nucleotide, and in the reference genome it could be 1 time, and in a person it could be like multiple times in tandem. So there are way more SNPs than structural variants, but structural variants are way more influential, again because they're much bigger, so their effects are much more detectable on the body. So where is the variation on the planet? Africa is the most diverse continent. The population has highest polymorphism and largest amount of private variants. So what is a private variant? This is a variant that is present only in the population of interest. So private African variants is polymorphism that exists only in Africa. So here you have the key. It's a pretty complicated pie chart of different regions on the planet. So the colors represent the continent. Blue is for Europe, like pink, magenta is for Asia, South Asia, and this is here in green for East Asia. And Africa is in yellow, and America is in red. And so you have the size of the pie represents the amount of diversity, like how many nucleotides are different, what's the percentage of nucleotide that's different within the population, and you've got here the different kinds of variation that are observed. You can see that this one here, which has the darkest color, is private population, private to population. So these are the variants that are present only in that population. And look in Africa, you can see that this sector here is typically bigger than if you compare it to, you got private to the population and to the continent. So it's both of those ones here. You can see that this is typically bigger than in Europe, but also typically bigger than even in Asia here in Africa. So most of the variation actually the variation is biggest in Africa, and that has to do with the fact that the human species, Homo sapiens, is actually coming from Africa. The population was there, so we have inherited the variation from there that was already present in the population. And as people have dispersed through the globe, going first to Asia, then crossing over to the Americas, some new variation has appeared, But most of it, most of the version that we see today is actually variation that we have kept from the very beginning, from the one that was in the ancestral African population. That is still true today, that just because of that, Africa is the most diverse continent. So SNPs and populations, the human population is structured at the genetic level because of the history of migrations. Sometimes this structure is spectacular. Okay, so like we have this initial pool of variation that's coming mostly from the initial population of Africa, but because people did not move much in the Middle Ages, then they just mated with their neighbors, and that means that that created pockets of variations that can be recognized very locally. And what you can see here is a map of Europe represented by the genes. The alleles, sorry. Left is a principal component analysis of the SNPs of European people. Arranging the SNPs by similarity reconstructs the map of European countries. Principal component analysis is a standard technique for analyzing high-level, high-dimensional data. And the gist is that 2 points are close if they represent individuals that have similar genomes, and 2 points are far if they represent individuals that have different genomes. So as you can see, when you just ask to represent the genome in a 2D space, so that you say place 2 genomes that are close together, close to each other, as simple as that. Then you recapitulate the map of Europe. Here this says all the people with a genome that looks like that, well, they're all from Spain, actually. And people with a genome that looks like this thing in pink, well, they're all from Switzerland. And people with a genome that looks like that in yellow-orange, they come from France. So it tells you that there is such a thing as a French genome, in a sense. Okay, like these are typical alleles that have a typical frequency that's observed in France. So it doesn't mean that most of the variation is explained by either races or by the geography. This is not at all what it means. It just says, and I insist, it just says that if I have your genome, I have a clue where your parents are from. It doesn't tell me how different you are from other people within that population, and it doesn't tell me that your genome explains most of who you are. It just tells me that there's enough information in your genome to tell where your parents are. And that's something that's very useful for forensics, so we're going to see in a second. I speak about that phenomenon on my blog, and especially on my blog I want to explain, go a little bit in more detail about how some of the arguments are used, let's say, to confuse people a little bit about what races are and what can be said about races and what cannot. So if you're interested about this, about the genetics of population, then you can have a look on my blog. So one thing that's important is that conservation is linked to variation. We want to conserve as many species on the planet as possible. This is something that we have to keep, because if we don't keep it, then we lose it. So it's as simple as that. Diversity is good for us. We have to maintain it. This is not something that we can lose, because if we lose it, it's lost forever. There are some cases of de-extinction of species, of course, but that's extremely rare at the moment. You have to be aware that this is still a dream, that we'll be able to resuscitate dead species. Most of the time when a species is gone, it's gone forever. It's easier to conserve them than to resuscitate them. So that's why many projects in the world are actually ongoing and using genomics to help conservation. And for those of you who are interested in doing something useful with genomics, it's probably one of the most useful things you can do for the world if you ever work in genomics. So that's an example here that I took from the literature. It happened a couple of years ago, and that shows what people are doing with genomics in order to help with the conservation and the variation. So the DNA from elephant tusks seized in Singapore in 2007 was compared to that of elephant populations in Africa, orange crosses. All the tusks matched a hotspot in Zambia, blue circle. The export point, red diamond, was several thousand kilometers north. Everything we know about illegal ivory trade routes is based on genomics. So I hope you understand the problem, right? Like elephants are being hunted by poachers, like this is illegal hunting, and the amount of poaching is not small. Some years the poachers have reduced the population by 20% of the world population. So if you have 5 years in a row like this, then elephants are gone. African elephants are gone. So poaching is not a small amount. This is a huge toll on the population of African elephants. Of course, they keep reproducing. The population grows again, thanks God. But these are not small numbers. These are huge numbers. And because this activity is illegal, it's very hard to trace. You cannot ask people, so how many elephants did you poach this month? Okay, 3, thank you. We'll get back in touch. This is all going underground. It's very hard to trace. So in 2007, the authorities in Singapore seized illegal tusks that were shipped from that point here in Africa, and using genetic techniques that we're going to see in a minute, they were able to say, okay, these tusks have the snips that correspond to those elephants. Okay, elephants, you can find them all over the place around here in Africa, but they said all the tusks, all of them, had snips that matched those elephants here. So that tells you that this is the place where they were hunted. They have traveled through other routes here to the port where they were then shipped to Asia, but it tells you that it was nowhere near that place here. It's actually from there. So that gives information now for the authorities to say that's the place we have to watch, because this is where they're the most active. There's no point putting that much watches around here, because anyway the poachers are there, that's for sure. And that's an information that you cannot get without genomics. Another thing that's quite interesting in terms of variation is ancient DNA. So DNA can be recovered from ancient bones. It is degraded, mutated, and highly contaminated by bacteria, and of course by human DNA, because we've been touching these things, and so now our DNA is on it. But with specific techniques, it can be sequenced. Left is the first bone remain discovered to belong to the human species called Denisovans. So here it's this bone here, the one that's here on the pinky, that tiny little bone of a child, of a very young girl from another human species. No kidding, they're humans, but they're not a species. The genome has now been sequenced. The species is now extinct a long time ago, but some alleles still survive in modern humans. And all we know about this species of humans, right? These are not races, these are different species here of humans that is gone extinct. All we have is just a little bone of a little girl, and being able to sequence her genome, we've been able to know exactly where they're from, the path that they followed. Why is that? Because some of the alleles of these species are still present in the population today, especially in Asia, if I remember, which means that allows you to trace a little bit the migration of populations, etc. And so an entire species of humans has disappeared, and all we have left is this little bone here, but we got the genome, and we can start to understand a little bit how people lived 20,000 years ago or more. Another thing that's quite interesting is that we can be focusing on what are the difference between individuals, how different are we to human from each other, but also we can start asking questions like how a human is different from a monkey, how a human is different from a lizard, or from, an octopus, or even a bacterium. So how would we do that? How would we be able to compare two different species? How would we even be able to say, oh, that piece of DNA here, or that cell, or that, that scale, whatever that is, any biological material, what species is it coming from? That's another question. So for this, we need a marker, a DNA marker, that would be spanning the whole tree of life. And it needs to have very demanding properties. So the first is that every organism must have it. Like, if you cannot find it, then you cannot do anything with it. It must be easy to purify, because otherwise you're going to lose it. It's going to be technically too difficult. It must be variable between species. Of course, if every species is exactly the same sequence, you won't be able to distinguish the species. That's kind of obvious. And it must be constant within a species. Yeah, of course. Now we want something that is really constant within one species, like all the members of the human population would have the same sequence, but monkeys would have a different sequence from the human sequence. So we're not looking for some things that are different among us, but that would be different between monkeys and humans, and still this, all the same within humans and all the same within monkeys. That's a lot to ask. And yet there is a perfect marker. And that perfect marker is ribosomal RNA. So that's absolutely in every organism except viruses. but it's a matter of definition whether this is an organism. They're easy to purify because there's a lot. There's really a lot. If you remember our class on RNA, you remember that ribosomal RNA is by far the most abundant RNA in the cell, and that there is already 10 times more RNA than DNA in the cell. So it's always the same sequence, and you got a lot of it. And not just a lot, you got tons of it. So that makes it super easy to purify. It's variable between species, indeed. So that's just an empirical observation that not every species got the same ribosomal RNA. And within a species, there's not too much variation because it's kind of core. Remember the ribosomal RNAs, they make sure that we can translate the proteins, and you can't mess up with that. Like the selection pressure on this are just so strong that the evolution is extremely slow. So with that, it was possible in the 70s already to get the tree of life. So using ribosomal RNA for phylogenies revealed the existence of a third kingdom called Archaea. So the person who did this, Carl Woese, found something that shocked the world at the time, is that he said, when I sequence the ribosomal RNAs of pretty much every organism I can afford to sequence, I can find 3 branches. Not 2 branches, not eukaryote and prokaryotes like everybody expected. I can find 3 branches. There are 2 branches of prokaryotes. So there's the bacteria here in purple, and that's the one that everybody expected, everybody knew. But he says there's also Archaea here in red. And in this brownish here, it's eukarya, like eukaryotes. So it was kind of a shock for people to realize that there is another kind of bacteria. They're not bacteria, they're Archaea, that are as different from each other as the bacteria are from us. The distance between bacteria and Archaea is actually huge. Of course, to us, they look like it's just like little microscopic rods, whatever that is. From the morphological point of view, they're exactly identical. But in terms of their biology, they've got nothing to do with each other. And so that was really a shock for the world to realize that maybe life is much, much more diverse than people had imagined, because morphologically it was not that diverse. And many people did not accept this idea easily, that there would be another kingdom, not just eukaryote and prokaryotes, but you also have like 2 prokaryotes domain, 2 prokaryotes kingdoms, and then 1 eukaryote domain. When you think about it, also if you're a little bit familiar with the techniques that I've been speaking about, DNA sequencing, the very first technique appeared in 1979, 1980. So this job was done before they could sequence DNA, or RNA for that matters. So this person, single-handedly working 10 years, was able to almost sequence the RNA using techniques that are absolutely, before any way of sequencing RNA could come about. So he used gel techniques, electrophoresis, etc. So all sorts of proxy for the sequencing of the RNA, and using that, he could still cluster the sequences together when they were similar. You can think about it like BLAST. Nowadays you compare sequences with BLAST. Like if the 2 sequences are similar, then they would match bioinformatically. But before that, what you could do is to hybridize the 2 pieces of DNA or RNA together, and you could see whether you get a good match or not. So with biochemistry, you can actually tell how similar sequences are without really having to sequence them. And still, after sequencing came, the picture did not change. It was not an artifact. People are now completely in agreement that there are indeed 3 kingdoms of life. And that's based on ribosomal RNA. Another cool thing that you can do with DNA is called DNA barcoding. And that's something that you can see the logo of Ontario here. That's something that comes from here. Like these are concepts that were developed here in Ontario, so they're not at the University of Toronto, but still this is a concept that's really coming from here in Canada. Some sequences used as barcodes include the cytochrome C oxidase subunit 1. So before I read that, I should explain what a barcode is, of course. So a barcode is a sequence that's very similar to ribosomal RNA. Like it has the same purpose as ribosomal RNA. So why would you not use ribosomal RNA if it's the perfect marker? Well, it's the perfect marker, but there are still cases where it's not exactly perfect. If you have 2 species that are quite close, for example, human and chimpanzees, the differences that you get between the ribosomal RNAs are going to be very few. There may actually not be any difference at all, because the evolution of ribosomal RNA is so slow then when the species are very close, you won't have enough differences to be 100% sure that these are actually different species. So you'd like to have a couple of backup sequences for that. You'd like to have sequences that are kind of varying enough between related species, something that would be human specific, and that also would be monkey specific, that would tell, okay, that's a chimpanzee, orangutan, or a human, or a gorilla, because each of them would have a different sequence. And if you use the ribosomal RNA for this, then it's not going to be that easy, because the differences are not going to be numerous enough. So that's the concept of a DNA barcode. That's a sequence that are kind of backup when our RNA is not sufficient, especially when species are close. So that's an example here of a particular sequence that's ideal. So you got an rDNA repeat here, and like this is the ribosomal RNA of one particular species. Like you can look at the DNA that's there, and that's the structure of a locus that's very often used here for creating DNA barcodes. Like within that DNA repeat elements, you can focus on some specific parts here that are useful for designing barcodes. So some sequences used as barcodes include the cytochrome C oxidase subunit 1 gene for all species, RBCL and MATK for plants, and the ITS sequence, internally transcribed spacer, for fungi. So what does that mean? It means that cytochrome C is an enzyme that we all have. Every organism that's alive has it, because it's an enzyme that's used for respiration in the mitochondria. So we all have it. And that's the same principle as ribosomal RNA. Like it's present, it's universal, so you can use it as a marker. Every species will have a different sequence there. RBCL and MATK are two plant loci. Every plant has them, but they have different sequences there. So these are loci or genes that are really plant specific. And the ITS, that stands for internally transcribed spacer, is this little sequence that I've highlighted here in the ribosomal DNA tandem repeats of fungi. So every fungus has it, and that's actually a very nice thing in the sense that you're not amplifying the ribosomal RNA, you're amplifying something that's inside the locus, because the 18S here and that 5.8S are very conserved, which means it's the perfect place to put primers for a PCR, but the ITS here in white is variable. Okay, so don't get confused, you're not amplifying the ribosomal RNA for making a DNA barcode. You're using the ribosomal RNA to design your primers because the sequences are conserved enough, so that then you amplify the variable region, which is highlighted in white. Okay, so what makes a good barcode sequence? It's something that has in yellow here a variable region that is variable between species, but that it's flanked by two constant blue parts here, so that you can have a pair of primer that will amplify this for literally every species out there. DNA barcodes are stored in the Barcodes of Life data systems, and that's an Ontarian database. That's one thing that Ontario is renowned for in the biological community out there. But again, not Toronto, almost. Another thing that has gained momentum in the recent years, especially in ecology, is environmental DNA, or eDNA. So eDNA is easy to collect. It can be used with barcodes to monitor the presence of some species in the environment. So what is environmental DNA? That's, there's a question that I missed. What is ETS then? Let me roll back. Up. ETS here is another variable sequenced that's there, and so that's the externally transcribed spacer, but this is not used for designing barcodes. Like they're using this here, this ITS there, and I'm not enough into fungus biology to tell you exactly why that is the case. So empirically this must work better than ETS, and NTS is non-transcribed spacer. So environmental DNA can be used to get kind of free barcodes. So what is it? It's like any sort of biological material that is shed in the environment by species. So here you have an example of fishes that are just releasing any sort of material, whether that be excrements or shedding scales or whatever that is, and it just goes directly in the water, and that kind of releases directly their DNA in the water. As simple as that. You can also have footprints sometimes, like there's enough of the material from the paw that you can collect in the snow, and then you get free DNA this way. Hair as well, like this is environmental DNA. But the easiest is really the one in the water. You just scoop the water, sequence the water, here we go. So what are the issues with that? It can be used to quantify the size of the population, but the stability of eDNA depends on many factors and is difficult to standardize. What do I mean? Is that the DNA, once it's released in the environment, will start to degrade. Okay, DNA is relatively stable, but think about it. There are DNases out there. The pH, the acidity of the water, the temperature will help catalyze all sorts of reactions that are going to degrade the amount of DNA. So just the amount of DNA that you find is not necessarily proportional to the number of individuals in the population. It is actually a product of how old is that DNA and how many individuals, were releasing that DNA in the population. So you can use it for ecology in order to monitor a population to see whether it's abundant or not, but it's really difficult to calibrate because you need to know something about the speed of degradation of that DNA in the environment. But if you know that, it's super easy. Like I said, scoop a bit of water, and now you've got your primers with your DNA barcodes, and you'll be able to identify the species that were somewhere in contact with that water. Simple. Okay, so it's time for the break. The second half will be about forensics, and we're going to see how to use more in-depth techniques to really trace criminals, etc. But for now, before the break, are there any questions, things you'd like me to explain again? Or is everything clear so far? I was going to ask about the whodunit assignment. Yeah, that's a good question. So right after this class, you will have enough knowledge to do the whodunit, and the whodunit will be released in a couple of days. So it's not ready yet. And the thing is that you will, again, each of you have a different exam. Okay, again, for the same reason, so that you guys can share tips and tricks, so that you can learn together, but that really you cannot copy paste the answer from each other's problems. You will all have different problems, but also different answers. So that will promote working together in order to understand, not to get the answer without understanding them. And the way it's gonna work is that I'm going to put the exams on the cluster. You will have to use your cluster account to retrieve the exam just for you, but it's not on the cluster. Okay, it's just a way to distribute 40 exams to every student that are different, but it's not going to be done on the cluster. And the exercises will be using techniques that I'm going to present in the second half now. I'm not sure that answers your question. Maybe you wanted to know more about the whodunit, like some more technical details. Okay, that was enough. Good. Any other questions about anything else? Doesn't look like it. So let's meet again. There is one question. So the assignment will not be a cluster assignment like the midterm? No, it's not going to be. You're done with the cluster. Okay, like I'm not going to ask you anything on the cluster. I will still use it to distribute 40 exams to you. Okay, because it's by far the easiest. Like you just download it and then you get the stuff this way. Otherwise, I don't have an easy way to write 40 mails to you. It's really not easy for me at all. And that's the reason. Is there some big barcodes specifically used for animals? Yeah, it's a good question. Like we've seen that ribosomal RNA is universal. Then the cytochrome is universal as well. We've seen the ones for fungi and the ones for plant. I'm not aware of barcodes that are really only for animals, actually, and I'm not sure why. The diversity of animals is much less than the diversity of plants or fungi or bacteria in the world, so maybe that's not much needed. Also, it's much easier for us to identify animals from their morphology. Okay, when you have a bone or even a hair, many experts are able to tell what that bone or hair is coming from. Like we know so much more about animals, but in terms of fungi, when you got a spore, they all look the same. It's just like little round. And even for experts in the field, like Yan Wang here at UTSC, even for him, it is difficult to delegate this species or that species. So DNA works much better in that case. So maybe there was just no need for developing barcodes for animals. I'm not sure. Will you have office hours this week? Yes, I do. Like normal office hour. You can book an office hour in person if you want. It's more difficult in person because I have to keep track, etc. And we have to keep 2 meters distance. So it's not fun at all to have it in person, honestly. But if you insist on having a office hour in person, I'll be in my office. The best is still to have it online. Alright, I think that was it for the questions. So let's have a 5-minute break. We meet again at 3:02. So have a little break, cool off if you haven't cooled off completely yet, drink something, and see you there at 2 or 3 past 3 o'clock. So is there anyone who is right now in the corridor, in the ones that are more or less in front of 311? Is this a good spot to follow the class, or is it really very difficult to follow the class from the corridor if you're online? I'm not sure if anyone is there in the corridor. Doesn't look like it. I hope that you guys managed to find a good spot if you were on campus. Library, yeah, library is of course a much better spot. Highland Hall, yeah. Are these good spots to follow the classes? Like it's not too noisy or you're not disturbing other people around? Yeah, good. At least that's good. All right, let's get started with the no. All right. Let's continue with the class. So now we're going to cover aspects of forensics. So the most important variation by far in terms of forensic is the short tandem repeat. So I'm going to spend a little bit of time re-explaining that concept that we've seen, I think, in the first or second lecture of the STR, or short tandem repeat. They have revolutionized genetic testing. They are easy to assay by PCR. They have many alleles, and they are very diverse. So what is a short tandem repeat? This is usually a tandem repeat of 3, sometimes 4 nucleotides. So tandem means that the repeats are really next to each other. Okay, they're not interspersed, they're really in tandem. So one unit, the other, etc. That's what's a tandem repeat. And short because they're like 3 or 4 nucleotides. I've explained the mechanism of expansion or contraction of these triplets of nucleotides. Is because when you have too many of them, at some point the DNA polymerase during the replication can kind of get full because you got these loops, especially around the Okazaki fragments, and so that can add or remove a triplet or more, because there are multiple ways of annealing the 3' end if it's inside these triplet repeats. So this is a really cool way to get some diversity about a genome, in the sense that the sequences on the sides, on the left and on the right, are constant, but the number of repeats in the STR are variable, and they're extremely variable, which means that every generation you have a chance that these repeats will expand or contract. But it's not a huge chance. We're talking of less than 1% per generation. But because there were just so many generations of humans already, it means that all of us have got like different alleles. And the good thing with other alleles, let's say, the things that you've learned in genetics, like with Mendel, is that usually you get 2 alleles. For a SNP, think about it. You either have one variant or the other nucleotide. That makes just 2 alleles. But with STRs, you get much more than 2 alleles. Like you got as many alleles as you've got number of triplets. So with just one locus, you would typically have around 20, sometimes even more, up to 50 different alleles. And that means that the diversity you're going to create with STRs is so much higher than what you could get with SNPs. So that's the sketch here. you're now very familiar how to design primers that would actually flank a region which is variable there, and you understand that the product you're going to get at the end is going to have a size on the gel that depends on the number of repeats. If you got many repeats, you got a big PCR product. If you got few repeats, you got a short PCR product. If the PCR product is short in the gel, it's going to migrate fast, so it's going to be at the bottom of the gel. If the product is long, it's going to migrate slow, and at the end of the migration, it's still going to be at the top. So with just one PCR, you can see which allele the person has. And that's a really simple assay to carry out compared to others. So this is particularly interesting for a system that's called CODIS20. CODIS20, or Combined DNA Index System with 20 loci, okay, Combined DNA Index System, is the FBI's standard for DNA fingerprinting. It amplifies sites on autosomes, each with 12 up to 98 segregating alleles. So that's the kind of stuff that you see in CSI and all these Hollywood-based, like forensic shows, etc. It's actually all based on the real system, which is called CODIS20. CODIS20 replaced CODIS13 that had 13 markers. So I think it happened something like 10 years ago, something like that. They put more markers than they had in the CODIS13 system, so they added 7, and now this looks more or less like that. You have the karyotype of a typical human with the chromosomes that are there, and each of these boxes here is the name of one of these sites that has been found by the FBI. And these are just STRs. Each of them is just a very simple plain LTR locus with a number of alleles that goes at least 12. Okay, 12 is the least variable, but that goes to 98 alleles present in the population. When I say segregating alleles, I mean that in the population among us, this is the variability that actually exists. They're not equally frequent, but you have 98 reported alleles in the population. And that's just one locus. And you got 20. So you can imagine now the number of differences that can be made with that system. So here you've got the boxes that are showed on the chromosomes. And very often they have this systematic name like D1S1656, like D1 because it's on chromosome 1, and then 1566 because it's like whatever number of short tandem repeat that was on that chromosome here. But sometimes you've got gene names like TPOX or FGA. So these are much older markers that were used in the 80s and the 90s, especially when people didn't yet have this systematic way of finding these STRs. So they were actually using genes. So these were the ones that were already present in the CODIS13 system. The one that have the systematic name were more added later in the CODIS20 system. You can see that some chromosomes don't have a marker, like chromosome 9, there's nothing on it, for instance. Some chromosomes have got 2 markers, but like they kind of span the whole genome, try to, to really cover the whole thing to maximize the coverage and the probability of getting variation that is really neutral. And it doesn't hit the X chromosome or the Y chromosome. Like they stay away from the X and the Y because that'd be confusing. So they have special techniques to assay the X and the Y chromosome, so it's on purpose that they're not putting anything on the X and on the Y. Okay, so that's the system that is used by the FBI, but also by other, other institutions on the planet to be able to tell what a piece of biological material, like who does that belong to. So you'd get a hair from me. You don't need to sequence the whole genome on the hair. You can just do 20 PCRs. You would get my 20 alleles 2 times, one that came from my dad, one that came from my mom, and then that is enough to give a fingerprint of who that hair belongs to. So you don't know who it belongs to, you just have the fingerprint. But if my DNA has been put in any database, you can make a match. You could say that person here has got exactly the right alleles that we have obtained by PCR. That has to be the person. The chance that it would be someone else is just way, way, way too low, because the number of possibilities of making combinations with 20 sites times 2 for the parents times, let's say, around 50 possibilities, it's really big. These are not times, actually, these are exponents in this case. So what about the gender? Sometimes you need to be able to tell the sex of the owner of the biological material, and because there was nothing on the X and on the Y, then it's kind of difficult to tell whether something with the codis system would be male or female. So actually you don't have enough information with just codis. For this, the typical technique is to use amelogenin, the amel gene or amel locus. So the amel gene is present on both X and Y chromosomes, but the Y version is longer by 6 base pairs. Okay, like there is an insertion on the Y, or you could say a deletion on the X, not sure. The sex of the sample can be determined with a single PCR of the amel gene. Okay, so like you've got the locus here. This is a very old technique, so like these are papers from the 80s already, and there is like a tiny little difference of 6 base pairs, which is here, between the X version and the Y version of the gene. So if you're male, you've got the X and the Y version, like there are an X and a Y in every male cell. If you're female, then you have twice the X version. So when you do a PCR, if you see 2 bands that are separated by 6 base pairs, it means that you've got a male sample. And if you see only 1 band, which is but double intensity, then that corresponds to the female version. So that's an example of how this looks like. That's positive control actually. So when you do the PCR on a female, you see a band which is like that. And when you do the same PCR on a male, then you see something that's like that. There are these 2 bands here, like the top band, which comes from the Y chromosome, this band here, which comes from the X chromosome. And you also have here this S or Y, so that's something else. It's a gene that's on the Y chromosome. So when you do a PCR on it, then you amplify only if it's a male. You cannot amplify anything if it's a female. So that's a control, and it shows that there is no signal here on female cells, and there is signal, of course, on male cells because they have the S or Y gene. So you will ask, but why not use the S or Y gene if it's such a good, such a good control? Actually, why use AML? I'm not sure if you can get the idea, but the main point is that it's not a good assay when you get nothing for one of the 2 cases. Okay, like if you get nothing with the SRY chromosome, SRY gene, sorry, it does not mean that the sample is female. It can also mean that your DNA was not extracted, that your PCR has not worked, or that anything didn't work, that there was a paracut during the PCR or something like that. So you don't want to have an assay in which when you get no signal, you will take a conclusion. You always want to have a signal. So that's why AML is a much better deal, because no matter what, you get a band, at least 1, sometimes 2. If you get 0 bands with AML, it means you did not extract enough DNA, the material was contaminated or degraded or something like that. So you have to get a band to conclude something. You cannot get an assay that is significant if you get nothing. That's the same thing that is used in the qPCR techniques, or the RT-qPCR techniques for SARS-CoV-2. There's always a positive control to show you have extracted enough RNA, you can actually get this amplification, etc. So assays which are based on band, no band are not really good. You want to have something that has either 1 band or 2 bands, because you always have signal. It's maybe not obvious for you because you haven't spent enough time in the labs really doing these kind of things, but for anyone who spent time in the lab, you recognize the value of this, that often the experiments just don't work, you don't know why, and you can't conclude anything. So that's the reason. It is not part of the codis 20, but it is often added to the fingerprint. So you would have actually 21 markers, like the codis 20 fingerprint, and then 1 more lane that would have the AML just to say is the fingerprints and the sex is male or female. So there's the National DNA Indexing System, or NDIS. NDIS is the database of the FBI associated with codis 20. It contains the profiles of many people, mostly offenders, and it is not accessible to the public. It is useful for partial matches. That's what we're going to see in a second. So codis 20 is the technique, that's really the methodology, and there's a database where all the alleles that they have caught with this codis 20 are put. That's called NDIS or NDIS. So don't get confused. One is the database, the other one is the technique. And so here you have the whole list of the 20 loci of codis 20 that I used here. There's a question in the chat. I'm going to answer it in a second. So you have the list of all the 20 alleles that I used in the codis 20, and you have here the number of alleles that exist for that particular marker, and you got two numbers, N1 and N2. N1 is the total number of alleles, N2 is the number of different sizes that can be observed. Because the codis 20 is not just different in terms of the size, which is like the number of triplets that you have, but sometimes these triplets have also mutated, which means that if you sequence the PCR product, you would even get an additional diversity. Like two people may have the same size of the STR, but they may still have a SNP inside the STR. So there was a question in the chat that refers to the slide before. What happens if someone is XXX or XXY? It's a very good question. Like, it's not frequent, of course, but that happens. So what do you think? XXX, you're going to have 3 copies here of the short version, so it's going to look like a female, because you're just going to get one band, which is the shorter band. XXY, you're going to have 2 copies of the short version and 1 copy of the long version. So it's going to look exactly like that. So it's going to be looking like a male, right? And if you have XXYY or these kind of other karyotypes, then it'd be the same. You get like 2 short versions, 2 long versions, still look like male, right? As soon as you've got one Y chromosome, you'd get the extra band, the longer one. And because you need an X chromosome to be alive, like it's not possible to just have the Y, then it would always look like either like this or like that. Well, that's a good question. Like aberrant karyotypes do happen. Sometimes they cause problems in this kind of analysis. So here you've got just an overview of the number of alleles that are possible for the different markers here, and also the fact that the typical number that they use is N2, the column that's labeled N2. They can actually get higher diversity by sequencing the product, and they get a couple more alleles in that case, and this is why they can go up to 98 different alleles in the case of the FGA marker that's here. So that database is not public. It's not like PubMed. You cannot just get a hair from me and then just sequence it with your newly acquired scale and then go to the MD system and say, let's see who this hair belongs to, and then get your person. Only the FBI and associated authorities can do that. That database is populated every time someone goes to jail or someone is convicted or is considered to be an offender in the US. They typically have to deposit not their whole genome, but at least their fingerprints. So they would take whatever biological sample they deem is appropriate from that person, typically maybe a blood sample, and then they would do the 20 PCRs on it, and then they would put that in the database saying, next time you do something, like it's going to be much easier for us to track you, because now we will just need a bit of your blood or hair or maybe any biological material that comes from you. We'll be able to say, okay, we don't need to find who that belongs to. We know it's that person because we already have their biological material in the NDIS database. So the way it works is that you can actually buy DNA fingerprint kits. So I never use them. This is not at all what we do in the lab, but I can imagine that this comes in a box and then you've got all the products. You just have to mix the things together with the biological material, put this in your PCR machine, and then you get the result. So DNA fingerprints are taken with commercial kits for multiplex PCR. The primers have a fluorescent label so that the markers can be distinguished. The assay is designed so that there is no overlap between the markers. So it's really very, very nicely designed. So you can do the PCRs together at the same time in the same tube. That's what's really nice. That's what is a multiplex PCR. They have optimized the primers so that they don't interact with each other. They will just each amplify their target sequence, and there are 20 of them, and that typically looks like that. So you have here a range of possible variation for that particular pair of primer, and then another locus, which is in this darker blue, with another possible range of variation here for that particular pair of primer. And this is really clever in the sense that the primers are fluorescent. There will be, for example, a blue fluorescent here in the 5' of that primer, and a green fluorescent label here on the 5' of this primer. So every locus that they amplify will give 1 or 2 bands. Okay, like either you homozygous and then you get 1 band, or you heterozygous, which is by far the most frequent case, and you get 2 bands. And these bands on the gel are actually colored. They have a color which corresponds to the fluorescent marker that was put on there. So they have 4 colors that they can put together, which are represented here by blue, green, black, not a color, or red, and black is of course a representation for the real color that's put on the primer. And so they use 4 colors, and also because they have made all this system being able to be multiplexed at the same time, like the places where they see the color each correspond to a different locus. So if it's something in this range here on the gel, so you have to imagine that this is going from left to right. Okay, like they have rotated the gel like this from left to right, and they're just representing the readout with the machine. They're not really showing the bands. But so when you read the blue channel, the one that corresponds to the blue fluorescence here, then what's in the range that is on the left corresponds to just one marker. Like there's no human that has any allele that'd be too much on the right or too much on the left. All the humans are within that marker. So with the blue channel, they can have, say, 4, you can see 4 markers at the same time. And so like each here of this row represents a different color which has multiple markers at the same time. So with just a few PCR in reality, you don't do 20 PCR, you do much fewer. And everything being automated, you get this gel with colored bands. You scan it with this machine that gives you this pattern here, and the peaks correspond to the alleles here. So they can directly tell for that marker here, which is hard to read as D8S1179, you have 2 alleles, one with 11 repeats, the other one with 13, I think. Okay, so you can imagine that they do that for every marker, and with just one super easy experiment, they get all the fingerprints from that person using this multiplexing techniques here. So that's typically how it's done. The results look like that. So what about the Y chromosome? Because CODIS is not doing anything on the X and on the Y on purpose. They can tell the sex with the allele, but sometimes you need to be able to do things, especially on the Y chromosome. This is actually a very useful chromosome for forensic. So when the offender is a rapist, it is sometimes difficult to distinguish the DNA from the stain and that of the victim, of course. Having specific STR markers on the Y chromosome allows the investigators to amplify only the DNA of the rapist. So you have biological material from the rapist, biological material from the victim, and of course there's much more DNA from the victim than from the rapist, as you can imagine. So if you do any PCR, any sort of PCR, you will amplify the DNA of the victim much more than you will amplify the DNA of the rapist. So that's why they left the Y chromosome alone, so that now you can have specific techniques that amplify only the Y chromosome. Because women don't have a Y chromosome, if you have primers on the Y chromosome, you're sure that you're going to amplify DNA from the Y chromosome, which is coming from the man, from the rapist in that case. So on the left, so that's the YSTR analysis is developed by this YHRD.org if you're interested in looking more about this particular set of techniques there. So on the left is the suspect SU that was released because its DNA was not a match ST. So you've got here the DNA from the rapist, which is this one that is, sorry, these are markers here that are controls. The suspect has a YSTR that looks like that, and the rapist here has this DNA. So it was clear from this type of analysis that the very first that came around, and it was actually used to say that the person was innocent, not guilty, because it said, look, like when we do the PCR on the Y chromosome of the rapist and on that suspect, it doesn't match. It cannot be the same person, right? So the person was released thanks to this DNA analysis evidence. Here there's just one band. That's a very old technique. This is not really what they're doing today. Today they use that many markers here. So we're talking about 6 or 7 markers just for the Y chromosome. So you can imagine you would have 6 or 7 bands one time, not 2 times. Okay, because the Y chromosome, you got only 1 copy when you have it. And so that tells you more about the identity of the person than just 1 band, of course. I hope it's clear here that the difference in the band says that if it's not the same band, then it cannot be the same person. So you have to look for another suspect. What if the rapist is non-male? This is a very good question. Of course, the vast majority of rapes are perpetrated by male to female, but it's not the only case. In that case, you can't use these techniques. It's much more difficult in these cases, and of course there are much fewer cases, so I'm not even sure that they're developing particular methods for that, and so I'm not aware what people would do. In that case, the the techniques, if the rapist is a female, is kind of hard. If the victim is male now, that's also another problem, and there are of course more cases of having a male victim than a female perpetrator. In that case, you cannot really use YSTR analysis, and I don't know what to do. If anyone actually has an idea or knew about this, now is the moment to suggest, because I'm not sure if there are some genetic techniques in that case. So, to understand YSTR, you have to understand the biology and the genetics of the Y chromosome, because it doesn't segregate the same way as the other chromosomes. The Y chromosome does not recombine. It just goes straight from father to son. Every man has the same Y chromosome as their father and their brothers. When you have a family, all the men have the same Y chromosome. It's not the same as the other chromosomes, okay? Because they don't recombine, they just inherited from the father, from the grandfather, from the grand-grandfather. It's just the same chromosome that goes down the line. Two brothers have got the same Y chromosome, exactly the same. This is not ideal for forensics with YSTR. There are new ideas to distinguish father's sons and brothers, but this is not yet standard. Okay, you see the problem is that when you got a match with the YSTR, you still can't tell which men from the family are the potential suspects, I mean the potential perpetrators, because they all have exactly the same Y chromosome. I have the same Y chromosome as my father and my brother. So the YSTR technique will just tell, okay, it's one of these guys, one of the Filion brothers, but we don't know which. And so there are the techniques, and so the idea would be to use hypermutable sites, so things that really mutate so often that even between brothers they would be different. Actually, you have a chance, which is approximately 30%, that there would be a mutation between my brother and me, for instance. But a 30% chance is not enough, if you see what I mean. Like, you're not going to convict people to a lifetime jail based on a 30% hunch. So you need something that's better than this, and that's why it's not standard yet. Another type of DNA that's used for forensic is mitochondrial DNA. So this one is the reverse. It goes on the female lineage. So the genome of mitochondria contains 3 hypervariable regions. They're usually labeled HV, so you can see them here, HV1, HV2, HV3, that can be used for genotyping. It is inherited only from the mother. Okay, the father doesn't contribute mitochondrial DNA. Because there are several copies per cell, it is easier to amplify than genomic loci. But if it has fewer alleles than STR-based fingerprints, therefore it's much less good. So as you're very well aware now, there are more than one copy of mitochondrial DNA per cell. And so just the fact that you got more copies of DNA of the mitochondria than the number of copies of the genome, it makes the PCR easier. If you see this, like you got at least 20, sometimes even more copies of the mitochondrial DNA than the nuclear genes. So for purification purposes, you're much more likely to obtain enough material compared to other to chromosomes, basically. So that's why it was very popular in the 90s, like because it was easy to purify, always give more signal than in the genome. The problem though is that you got only 3 loci that are variable, and so it's much less than the code is 20, for instance, and each of these loci doesn't have so many alleles, so you would be able to distinguish, for example, 2 people that live very far from each other on the planet, an African versus an Asian, for instance, because there would be enough time for these hyper variable regions to mutate. But if these people are from the same region, like there would not be enough variation to distinguish 2 people. So that's why nowadays they don't really use mitochondrial DNA anymore as much as they used to in the past. Like the coded system is considered to be much better. Like you have much more variability to identify a single person. Another thing that's important to realize is that deep learning is making really a strong point in the fields of forensics. So there are two problems that deep learning is promising to solve, and so far the results are so-so, but people are quite hopeful that it will improve in the future. The first is called DNA admixture. You can see the profile that is here. So that's a Codis 20 profile. You can recognize them now, but you see that there are more than 2 bands every time. You got like 4 bands here, 4 bands here, and maybe a half, etc. So it means that it's not just one person. You got, let's say, there's the blood of one person and the blood of another person that have been mixed. You can't distinguish them. So when you do the PCR on them, then you get the DNA from both people. Or that's the case that we just mentioned about rape. Like you would have like the DNA from both people, and you have to kind of know which belongs to which. So DNA admixture means that several people contribute to a profile. Some claim to be able to separate the profiles using these deep learning techniques, like these new machine learning techniques, to be able to distinguish these two profiles, to say, okay, there is that person which has this fingerprint and this person who has other fingerprints. Otherwise, you got a mix, which is very difficult to distinguish there. And another thing which is making kind of rising a lot of interest but also fear is DNA facial recognition. So here that's an example of a person who was whose DNA was sequenced, and they asked the machine, tell me what this person looks like, and the machine came up with that version of the face of the person, and that's the true face of the person. And you can see that it's not the same, but it's kind of looking close by now. So DNA facial reconstruction is making fast progress, but it is not based on biological understanding. So I want to highlight that it doesn't really go at all with the idea of understanding how the cranial genes are set in place during development, etc. It's just based on statistical similarities. So you just show pictures of, like shots of people, and then you show their genes, and it just says, oh well, okay, that person probably I'd say the skin is black because they've got this SNP that's actually very often found in populations where people are black, but it's not because there is any causal link between that SNP and the skin color. And I say, okay, the eyes are blue because this is a SNP that's very often found in the north of Europe where people have got blue eyes. And also I say that the hair is black because whatever, like these things are all based on statistical criteria, but this is not at all based on understanding how the genes are causing the body to look this way. That's a little bit confusing, that this is purely statistical. There's no biological understanding whatsoever. Still, it works, kind of. This is one example. It's actually taken from the website of the company that develops this. So of course they will showcase the one or two few cases that are extremely good, but there are also plenty of cases where it's not that convincing. In particular, a person can have shaven the head, as simple as that. You don't even know the age of the person. The person could be like 50 years older than you think. So just to bet that the person would be obviously between 20 and 40 is already a bet that is not in the DNA. So definitely there are going to be some applications coming from that angle, but we have to be reasonable. Not everything is in the DNA, and we have to keep a little bit that in mind. And also there are some gadgets, like funny things. One of them that I found particularly funny is DNA spray. So what is DNA spray? The DNA spray technology is used to spray a known sequence that covers body and clothes. So it's really spreading primers, basically DNA primers that are easy to synthesize. It is hard to wash off and can be used to confirm, for instance, that a suspect was in a bank when he was robbed. So this is in action in the Netherlands. So here it says this shop is being monitored by DNA spray, and so like it means that that's a bank. You come, you rob the bank, say give me the money, nothing happens. But while you do that, there's an invisible spray that just puts this PCR primer all over the place, and that goes inside your hair, that goes on your skin, etc. And it's just because it's hard to wash. Like you can try to wash it with soap, but DNA is quite resistant actually compared to other molecules. And because PCO is such a sensitive technique, you just need one molecule to have remained on your skin, and there'll be evidence enough that yeah, you were in that bank at that moment, because that's the only time in the world and the only place where there has been this spray. So if we can PCR this thing off your skin, then you're probably in trouble because you were in that bank at that moment. The other thing is that people are afraid of DNA in the Netherlands. So just having this thing is more of a deterrent than anything else. They think like, oh my God, DNA doesn't look good for me. I'm probably going to stay off that bank and rob another bank that doesn't have this sign in front. And here that's just a kind of a hacker advertisement on the internet showing that DNA is fluorescent, of course, and so they're making the point that this person's face was covered with the DNA that was in the bank, and you could see the DNA this way, but that's not at all the way it works. Alright, so that's more of a gadget, but that tells you how people are inventing kind of techniques based on the fact that DNA is such a unique molecule that you can identify so easily with PCR, so you can use it as a tracer, as a marker. That's all I wanted to say about forensics. You can see that there's a lot of very interesting things happening right now. Genomics is a very exciting kind of technology for people who are interested in this. And the key points to remember from today's class is that diversity can be used to identify things. Okay, like even without understanding the biology, where it comes from, what it does, there's enough information out there to say this sequence belongs to that organism. STRs are the standards for individuals. Okay, the short tandem repeats, and in particular the coded system, is really the standard technology to be able to identify one person in the whole population, one individual if we're talking of plants or animals. And for species, it's DNA barcodes. It's the standard to be able to say, okay, it's that species compared to that other species. So now it's time for the breakout group activities. Is there any questions before we go into breakout rooms? There's plenty of time today for the breakout exercises. You'll see they're pretty fun today. Any questions about what we've just seen? Doesn't seem like it. So let's go into the breakout. No, there's a question. How do we deal with people who carry two different genomes, like after a transplant? Well, it's a very good question. You have to think where the transplant is, for instance. Imagine that this is a heart transplant, something like that. In that case, there's very little chance that you'd have some heart material that is coming, in the hair or something like that. Okay, if it's the blood, you may have a little bit of DNA coming from the heart, of course, but most of the DNA will come from the rest of the body, from where the blood is actually from, where it's where the cells are made, and it's not in the heart, it's in the bone. And so I think in most of the cases there will be an overwhelming majority of the DNA coming from the original body, let's say, and a tiny portion that will come from the external organ, from the organ coming from another person. So my expectation is that it would not be a very big deal. Like you get like one major peak that corresponds to the major body, and a tiny peak, if anything at all, coming from the other organ. It'd probably be detectable, but you still be able to distinguish which comes from which. But it's just a guess. Any other question? All right. So here are the exercises. In the first exercise, you'll have to work with HIV, intentional contamination. A lady who has contracted AIDS accused her MD boyfriend to have injected her with HIV contaminated blood. The would-be source patient was found, and the two HIV were partially sequenced. So you have the DNA here of the HIV from the lady, the DNA here from the HIV from the patient that she claims she got the virus from, and you have reference 1 and 2. There are just two other HIV patients picked at random out there in the wild in North America. So see the HIV sequences of the lady, the patient, and the two unrelated reference patients with HIV. What do you think happened? And here's a hint here. So you have to tell whether you think that indeed the MD boyfriend was cruel enough to do that, whether the lady is kind of way over, I don't know what, and she's inventing that stuff. A cold case. Several women in the west coast of the US were raped and murdered in the 1980s. YSTR methods showed that the rapist was the same, but he did not make more victims, and he was not caught. In 2018, a 32-year-old convict gave a DNA sample with a perfect YSTR match. Who was the rapist? What did the FBI do next? Criminal mood today. Third exercise, a beautiful story. A man was found with a conserved specimen of endangered butterfly in his car. He claimed that he acquired it legally from another continent. The pollen from the proboscis of the specimen was collected, and RBCL barcoding was performed in search of Calypso bulbosa, a flower that grows exclusively in the region, not on another continent. 3 barcodes were found, like 1, 2, and 3. What is your conclusion? And here's a hint that you can access the BOLD database at that link here and see what happens there. So that's it. That's the program for today. I'm going to stop the recording. So thank you very much for those who are watching the recording, and you will see the answers in the written form of the lecture.